I'm looking at experienced forager honeybees and then less experienced, newly emerged honeybees, and seeing the difference in this certain region of their brain called the mushroom bodies, which has been connected to learning. Our brains have neurons in them that communicate with each other through neurotransmitters, which are these chemical signals. These chemical signals can either excite neurons or inhibit neurons. We're specifically looking at inhibition and what role this inhibitory neurotransmitter or signal called GABA plays in forager honeybees and their process of learning.
There are categories of humans for all sorts of reasons who suffer from intellectual disability. And surprisingly, that's sort of regarded as an incurable condition. Modern neuroscience thinks brains normally are changed by experience. We can figure out how to make brains of persons with intellectual disability maybe function a little better. Honeybees are a nearly perfect model. And so I've spent a lot of time trying to figure out what changes occurred in the brain, and we track them down at the cellular and molecular level, and then how having a bigger part of your brain is important for improved behavioral performance. I met Naoki. He was a student in my introductory biology class, and he had a lot of good questions. And he approached me and said, I want to become a neuroscientist. I want to teach neuroscience, think about neuroscience, but I don't know what I want to do. And I said, we got to start somewhere. So started my lab.
I had never gotten any prior lab experience in high school, and I'm actually going into a lab in the fall, and I kind of wanted something to prepare me for that so that I'm not just completely thrown into that setting with no idea what to do. And so this URECA program provided a great avenue for that. I've always been interested in learning and memory in particular. I have long-term goals of studying Alzheimer's and dementia, and so that's always been an area of interest. Being completely new to a lab setting, I was really, really nervous for this experience as well, as much as I was excited. And I think Dr. Fahrbach was a great mentor for me because she's very understanding, very calm. I think one major takeaway that I took from the lab is that every step leads to the next, and that the care you put in to one step is going to be fruitful for the next one. And so it really made me pay attention to whether it's by sectioning or in the immunolabeling process. Every single detail of how you do something makes it easier or plays a larger role in something that comes downstream. And I think that's a really, really valuable lesson that I'm going to take into future experiences as well.